All right, so welcome to this week's lab. Unfortunately, I can't spend it with you live. I've got to do a recording of it. So we're going to go through Lab 8, Photosynthesis, the procedures, safety instructions, and the general content. You guys are going to be on your own for actually running the experiment and finding the materials. Please note that this week will require you to pick up several materials from the store, so make sure you have those before you sit down and do them. Make sure you do this, well before the due date of the assignment. This week we're talking about photoautotrophs. These are organisms that transform the kinetic energy of the sun's light into the potential energy of the bonds of glucose. It's a transformation. The photoautotroph we're going to be looking at are plants. Plants have structures in them called chloroplasts. This is where photosynthesis occurs. Specifically in the chloroplast, photosynthesis is going to occur in the thylakoid membrane. So if you look at the chloroplast, it's going to be this circled structure here, these flattened discs within the chloroplast themselves. Now, as we recall from lecture, light in plants is going to be absorbed in the red and the blue spectrum. That means that we're getting energy. Plants get energy from light in the blue section, blue range, and light in the red range. It does not absorb light in the green range. In fact, it reflects that light. It cannot use energy from green light in order to conduct photosynthesis. That's why plants are green. They're reflecting that light. Chlorophyll is that green pigment. It's embedded in the thylakoid, absorbing light. That energy then energizes an electron. It makes that electron ready to do work. And the work the electron is going to do, and this should sound familiar, is go step by step by step to push hydrogen across across a membrane to form an electrochemical gradient. And that electrochemical gradient of hydrogen ions is going to flow through ATP synthase to produce lots of ATP. That ATP, in turn, from the photosystems, is going to be used during the Calvin-Benson cycle in order to create sugar, glucose. So all of the ATP, all of the NADPH that was built during the photosystems gets shifted over into building sugar in a three-step process called fixation, reduction or carbohydrate production, and regeneration, because you're regenerating RUBP, ribulose bisphosphate. You'll notice that there's this major enzyme here. It's called ribisco. Ribisco's job is to take carbon dioxide and take RUBP, ribulose bisphosphate, and combine them together into a larger molecule. It fixes carbon dioxide. That's why it's called carbon fixation. You're taking the carbon dioxide and affixing it or gluing it to another molecule. Then that RUBP goes through a process where it's reduced. That is, it's going to gain electrons and eventually get built into glucose. Next, and finally, you have to use up the rest of your energy to rebuild RUBP so the process can begin again. So we're going to assay photosynthesis. That's this week's lab, assaying photosynthesis. We're very interested in how do different colors of light impact the rate of photosynthesis. Now, we just talked about what colors of light are absorbed and what colors of light are reflected. White light is composed of all the different colors combined together. So we're going to look at white light as our control. We know that it works. It's a positive control. We're going to look at red light, and we're going to look at, I believe, green light. One of those should work. One of those shouldn't. It's up to you to figure out which is which. In this research, we're going to need to figure out what our dependent variable is. So on the screen in front of you, you can see that you've got carbon dioxide, water, and water come together in photosynthesis to form sugar and oxygen. So you can measure a bunch of different things. You can measure how much carbon dioxide is being used up. That is, how much carbon dioxide are you losing? You can measure how much water are you losing, how much sugar is created, or how much oxygen is created. Those are your different potential dependent variables. For us, we're trying to make this as simple as possible. We're going to use aquatic plants, the ones that live underwater, in order to cause photosynthesis to occur. These plants use carbon dioxide, but they release oxygen. And they're going to release oxygen in the form of bubbles. For us, we got to give them some carbon dioxide, though. So in the solutions that you're preparing, you're going to have baking soda. Baking soda breaks down in water and releases carbon dioxide. So that's where we're going to get our carbon source, the baking soda. Between that and some carbon dioxide and a little bit of water, we're going to be able to have the plants undergo photosynthesis to produce oxygen and glucose. So get all your materials together. You're going to need 8 clear cups, preferably plastic so that nothing breaks, tap water, some sharp scissors, a stopwatch, some measuring spoons. You're going to need natural sunlight or a 60-watt bulb, what's called a goose lamp. You'll also need an aquatic plant. That aquatic plant may be difficult to get, so you got to do some searching. You're looking for either Elodea or an Anacharis. These are commonly available at local pet stores. You'll buy a small bunch of them. They are extremely efficient at photosynthesis underwater because that's what they evolved in. If you cannot find these plants, you can substitute just baby spinach, which you can find at any grocery store. The baby spinach will not work as effectively. It may be much more difficult to do, and you may need to run this experiment for longer to see the same reaction. It is ideal to get either Elodea or Anacharis. If not, spinach will work, but it's going to take you about twice as long to run the experiment. So we need to first prepare our photosynthesis chambers. First step, measure out 500 milliliters of tap water into a beaker. Add about a half a teaspoon of baking soda. Now remember that baking soda's job is to release carbon dioxide in the solution. You need to stir it up so you have carbon dioxide for your plants to photosynthesize. Label your cups. One will be clear, one will be red, one will be green. You'll see why in a second. Then divide your baking soda solution. So you've got a bunch of baking soda solution. Divide it evenly into the 3 cups. Add 3 drops of red food coloring to the red cup, 3 drops of green food coloring to the green cup, and leave the other cup as clear. All right, so 3 cups are done with baking soda solution, a red one, a green one, and a clear one. Next, you need to take a 4th cup and label that as your control. That 4th cup is going to just have water in it, no baking soda. Okay, if we don't have baking soda, we don't have carbon dioxide, we expect there to be a low level of photosynthesis occurring. Next, you need to prepare your plant. So you should have Elodea or Anacharis. It's a long plant. You'll take a sharp pair of scissors and cut that plant at an angle. So you don't want to cut straight across the stem. You want it to be cut at an angle on the stem. Once you cut that angle, you crush the stem. You don't want to, basically a stem, the whole job of the stem is to move water through that plant. You want to hamper that as much as possible. You want to close it off. Then you take your cut plant, about 1 inch of your cut plant, and you put it into your test tube. Don't push it all the way down to the bottom and crush it against the bottom. You want to push it down to the bottom, use a pencil or a pen, whatever you need to in order to get it down. And then you want to not have it at the very bottom. It needs to be a little bit higher off the bottom so it's not touching it. And then you can move on. You're going to pour water from your control tube into the from your control cup into the test tube until the very, very tip of the rim. Then, while holding the plastic, you're holding the test tube in one hand, plastic cup in the other. You put the plastic cup on top of the test tube. You put the test tube up so that the water level of the test tube is touching the surface. That test tube should be filled all the way to the brim, and you've got water at the tip. And then you take that whole thing and you flip it over. Label that cup as your control and move on to the next. Repeat that process for the red, green, and clear cup. On the next slide, I produced a video for you showing you how to do this because I don't know that the words are good enough. Even my little hand motions here. Sorry about the quality of this video, but I thought that you could use a video to do this instead of and trying to interpret what it says in the lab manual.
basically, what I'm going to do is I've got my solution prepared. I'm going to pour it into my test tube. Solution, I colored them so you can see the solution moving, otherwise it'll just look clear. I want to fill up this test tube as much as I can. You have your test tubes in your lab kit. And you'll notice at the top of this test tube, I've got a little bit of space. So I'm going to take my my transfer pipette. I want to get it all the way to the tip top, all the way up. Now, once I've got it all the way to the top, I want to take my plastic cup. I'm using a beaker, but you'll use a plastic cup. Put it on top, give it a little hat, and get it so it's flush against it. So you don't want, you want the, it's dripping a little. So then I flip it over, and most of that liquid's still in there. Take the remaining portion and pour it in. Just like that. And as you can see, now the test tube is in there and it's holding the water in it.
Next, you're going to run the experiment. Take those cups that have your inverted tube and your plant in it, and you want them to be in direct light. So that could be in direct sunlight, or it could be indirect light underneath the lamp. Leave the cups untouched for about 20 minutes. That lets them equilibrize. That lets them get used to their new conditions. After 20 minutes, set a timer and watch how many bubbles are produced in one test tube at a time. Count the number of bubbles that are produced, either on the leaf or that float up. Repeat that count for 2 minutes, and then do that 3 times. You average the number of bubbles produced, and that should give you your best estimate as to your data. All of that data will be recorded on table 8-1 in your lab book. You'll also need to take a picture of your setup at this point. That goes on your lab handout. Hopefully so far so good. Once you've got your average number of bubbles for clear, control, red, and green, you're going to graph them. You're graphing only the average number. Do not graph each individual one, just the average. And remember, your graph needs to have a title. You need to have your independent variable on the x-axis, and it needs to be labeled. In this case, I don't want just like tube 1, tube 2, tube 3, tube 4. I need to be able to look at your graph and know exactly what you're talking about. Then your dependent variable goes on the y-axis, the number of bubbles. It needs to be clearly labeled. Safety notes. The biggest safety note here is to be careful not to slam your test tube into the table when you're flipping it. It can be tempting to be very aggressive in that flip, and you could break your test tube. You don't want to do that, especially while you're holding it. Also, as per normal, please don't drink the solutions. Baking soda won't kill you, but it's also, well, actually in high doses, but just don't drink it.